So children, next we will discuss about the tissue culture. Now what is tissue culture? Tissue culture means that when we take a small portion of the tissue or organ from the parent plant, and when we culture this small portion in the culture medium, then we will able to get in large quantity the tiny plantlets, and this is known as the tissue culture. That from a small portion of the tissue and organ, how we can get thousands of the plantlets, and this is also known as the micropropagation. Micro means small portion, and from small portion thousands of plantlets. So let's discuss in detail that how tissue culture that is performed. So first of all, a small portion of the tissue or organ that is extracted from the plant tissue, or it can be organ, and this is known as explant. Now this portion that is cultured in the culture media, in the Petri plates. These are Petri plates, and in the Petri plates, this culture medium that is present, and culture medium, it contain agar that is the jelly like substance which contain the nutrients, the required nutrients for the growth, and plant hormones. Now this tissue or organ we have to take in the culture media. Now what happened? By using the nutrients or the plant hormones, this tissue having the cells, they will divide rapidly and form a lump of cells, and which are unorganized. Okay, and these are known as callus. As the cells is present in the explant, they divide rapidly and form the callus. Callus, it contains the unorganized group of cells. Then after that, what happens? When the callus formation will be there, then the medium that will be transferred. Means this callus, then it will be shifted to the another culture medium containing the plant hormones which are responsible for the root formation. Okay. Now this callus that is transferred, it is transferred to the culture medium containing this culture medium. It is containing the plant hormones which initiate root formation. Okay. So now what happened? The roots, they will arise from the cells present in these tissues or organs. Now again these cells that are transferred to the another medium, another culture medium, this medium that contain the plant hormones which are responsible for the shoot formation, which initiate shoot formation. Okay, so now they look like this. So when the roots as well as the shoot, they will arise from the cells, then what happens? They will become the tiny plantlets. They known as the tiny plantlets. Now these tiny plantlets, they are separated from the culture medium and then they are transferred either to the soil in the field or in the pots, so that they will get enough space for the growth and development. Okay, this is soil in the field, means it can be transferred to the soil that is present in the field or in the garden or even in the pots, so that they can get enough space for the growth and the development. So in this manner, thousands of plants that can be generated from a small portion of the X plant, and that's why it is also known as micro propagation. Micro propagation, and that's why this is known as tissue culture, because from the tissues or organs the plants that are generated. Now what are the advantages of the tissue culture? Tissue culture, it can be performed even in a lab. Okay, so as it can be performed in the lab, so it requires very less space. First, it requires very less space and it requires very less time also. Okay, and it is irrespective of the weather or season. If suppose rainy season or sunny season, means it is irrespective of the season, you can grow it round the year. Either it can be rainy outside, it can be sunny outside, because you have to grow the initial stages in the lab. And the next thing is that as you take the growing tip, means you take the X plant from the growing tip region, so that's why that tip that contain the meristematic cells, and as the tip that contain the meristematic cells, and you take the meristematic cells for this tissue culture, so it is also diseased free. Means the plants you grow from the tissue culture that are diseased free plants, because the meristic cells, the meristematic cells, they are protected against the pathogens.